Cell membranes are made of a double layer of lipids called the phospholipid bilayer. This special structure contains innumerable phospholipids with polar heads composed of phosphates attached to other ions and polar molecules, and nonpolar tails composed of long fatty acid chains that repel water and thus gather to form the inner layer of the membrane. The interaction of the heads with water and the grouped tails allows the membrane to be fluid. Phospholipids can easily drift around within their own layer and rarely even flip to the other side. How easily phospholipids move around is membrane fluidity. Too fluid and the membrane won't hold together. Too viscous and the essential life-giving properties of the membrane, like allowing proteins to move around within the membrane and interact with each other, would stop or slow down. Having the right consistency is key, but what affects it? We're going to focus on two properties: saturation of the fatty acids and cholesterol content. Saturation of the fatty acids is actually pretty straightforward. More saturation of hydrogens and you create a long, skinny, straight fatty acid. An unsaturated fatty acid with cis double bonds creates a bend. Polyunsaturated fatty acids with many cis double bonds can even create curves like this omega-3 fatty acid. Ever try to pack a crumpled up, paper back into a folder or a binder? You just can't fit as much as you could if it was lovely and straight or flat. Similarly, double-bonded unsaturated fatty acids open up space in the membrane, allowing it to be more fluid. This is an important property of membranes in animals that are cold-blooded and living in cool or cold habitats. That's why we get a lot of our omega-3s from fish oil. So to recap, the more saturated the fatty acid, the more viscous the membrane. The more unsaturated fatty acids, the more fluid the membrane. Cholesterol has a stabilizing influence according to temperature. When temperatures are moderate, think human body temperature, cholesterol reduces fluidity and stabilizes the membrane. When temperatures are cool and fluidity tends to decrease, cholesterol actually prevents the fatty acids from packing tightly together. So at cold or cool temperatures, cholesterol increases or promotes fluidity. Or, to put it another way, cholesterol lowers the temperature required to solidify a membrane. Pretty cool? I think so. There are other minor influences at play. Longer fatty acid tails and overlapping tails will both reduce fluidity as the tails can catch on the opposite layer. But saturation and cholesterol are the two biggest influences to membrane fluidity.